The Krebs cycle, understanding the biochemical pathway of energy production. Hello, welcome to the Science Guide ASAP Learning. Today we are going to be speaking about the Krebs cycle. Hope this helps. Please support this new channel by liking this video and subscribing for more videos like this one. The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid, TCA, cycle, is a fundamental metabolic pathway that plays a critical role in the production of ATP, the primary energy source of the cell. The cycle occurs in the mitochondrial matrix of eukaryotic cells and is a series of 8 chemical reactions that convert acetyl-CoA, a molecule produced from the breakdown of glucose or fatty acids, into ATP. In this video, we will explore the steps of the Krebs cycle in detail. Step 1. Formation of citrate. The Krebs cycle begins with the binding of acetyl-CoA to a 4-carbon molecule called oxaloacetate. This reaction is catalyzed by the enzyme citrate synthase and results in the formation of a 6-carbon molecule called citrate. Citrate is the first intermediate in the cycle and is also known as citric acid. Step 2. Isomerization of citrate. Citrate is then converted into its isomer, isocitrate, through a reaction catalyzed by the enzyme aconitase. Isocitrate is also a 6-carbon molecule and is the second intermediate in the cycle. Step 3. Oxidative decarboxylation of isocitrate. Isocitrate is then oxidized and decarboxylated to form alpha-ketoglutarate, a 5-carbon molecule. This reaction is catalyzed by the enzyme isocitrate dehydrogenase and results in the production of NADH, a molecule that will be used in the electron transport chain to generate ATP. Step 4. Oxidative decarboxylation of alpha-ketoglutarate. Alpha-ketoglutarate is then oxidized and decarboxylated to form succinyl-CoA, a 4-carbon molecule. This reaction is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase and also results in the production of NADH. Step 5. Formation of succinate. Succinyl-CoA is then converted into succinate through a reaction catalyzed by the enzyme succinyl-CoA synthetase. This reaction results in the production of ATP through a process called substrate-level phosphorylation. The ATP produced in this step is the first ATP produced by the Krebs cycle and is also known as GTP in some organisms. Step 6. Oxidation of succinate. Succinate is then oxidized to form fumarate through a reaction catalyzed by the enzyme succinate dehydrogenase. This reaction results in the production of FADH2, another molecule that will be used in the electron transport chain to generate ATP. Step 7. Formation of malate. Fumarate is then converted into malate through a reaction catalyzed by the enzyme fumarase. Malate is a 4-carbon molecule and is the penultimate intermediate in the cycle. Step 8. Oxidation of malate. The final step of the Krebs cycle involves the oxidation of malate to form oxaloacetate, the starting molecule in the cycle. This reaction is catalyzed by the enzyme malate dehydrogenase and results in the production of NADH. History of the Krebs cycle. The Krebs cycle was first discovered in 1937 by Sir Hans Krebs, a German-born British biochemist, and his colleague William Johnson. Krebs was studying the metabolism of pigeon breast muscle when he discovered a series of chemical reactions that eventually led to the production of ATP. He later named this process the citric acid cycle, and it was later renamed the Krebs cycle in his honor. Regulation of the Krebs cycle. The Krebs cycle is regulated by several factors, including the availability of oxygen, the concentration of ATP, and the presence of inhibitory molecules. The rate of the Krebs cycle is directly proportional to the rate of oxidative phosphorylation, which is the final step of cellular respiration. Inhibition of this process can lead to metabolic disorders such as lactic acidosis. Diseases associated with the Krebs cycle. Mutations in genes that encode enzymes involved in the Krebs cycle can lead to metabolic disorders such as mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, MELAS syndrome. These diseases are characterized by impaired ATP production and can lead to a variety of symptoms, including muscle weakness, seizures, and developmental delays. Current research and future directions. Research on the Krebs cycle has led to significant advances in our understanding of cellular metabolism and the treatment of metabolic disorders. Recent studies have focused on the regulation of the Krebs cycle and its role in the development of cancer. Future research may lead to the development of new therapies for metabolic disorders and the prevention of cancer. Overall, the Krebs cycle is a highly regulated and tightly controlled pathway that plays a critical role in the production of ATP. The cycle is regulated by several factors, including the availability of oxygen, the concentration of ATP, and the presence of inhibitory molecules. Understanding the steps of the Krebs cycle and its regulation has significant implications for our understanding of cellular metabolism and the treatment of metabolic disorders. Hope this helps. Please support this new channel by liking this video and subscribing for more videos like this one.